Today we are going to discuss about cystode infections, that's tapeworm infection. So moving on to the specific objectives, these are the objectives given in your module book, which I have just copied and pasted, but we'll move on to the intermediate objectives for this lecture. So let's see what are the intermediate objectives. At the end of this lecture series actually on cystodes, you should be able to list the parasitic infections, that's cystode infections, cystode worms causing gastrointestinal tract diseases in humans. And also you should know their morphology, the life cycle, and how they are being transmitted to humans. And also you need to know the clinical aspect, the clinical manifestations caused by those parasitic infections, as well as the pathological impact by those parasites to the human body. In addition, later on you need to know how can you confirm those parasitic infections in the laboratory. Now you first get their history, the clinical examination, they will give you a clue to which parasitic infection this is, and then with the help of laboratory diagnosis, laboratory test, you are going to confirm the diagnosis. And in certain instances, there may be radiological investigations which can be done to diagnose certain parasitic infections. That also you need to know. Finally, you need to know the principles of management. That means, now here in the lecture, you are going to learn the principles of management, kind of the medical management or the surgical, what kind of management aspects you are going to use to control or treat this patient. Now, the real management, of course, you will learn when you go for your clinicals. And finally, you need to know the prevention and control with little bit of epidemiology. Of course, epidemiology changes from time to time, but right now you are going to learn whether this disease is prevalent in Sri Lanka and what is the impact that it has caused to the Sri Lankan community, and then how do you prevent and control this disease. Now, before I move on to the next slide, I would like to say that list is, when you are asked to list something, just name them. No need to go into further details. But if you are asked to describe, you need to go into details. You need to know a little bit of more details, like you need to know deeper in the particular topic. The discuss is, on the other hand, is you are discussing the advantages, disadvantages, and different aspects of the particular topic. So those are the three different things that we have used in the intermediate objectives. So you need to know those things separately. This is the lecture outline. I'll be giving you an overview as to what we are going to discuss in this series of lectures on cystode infections. Basically, I'm going to tell you where the cystodes lie in the classification. And then we will discuss what are the medically important cystodes overall. And then later on, we will concentrate more on the species causing cystode infections in Sri Lanka. And later on, at the end of the lecture, I'll be talking about the general morphology of cystodes. Moving on, now you have already learned about roundworms. Now, during that time, you would have heard this word helminths. Helminths means worms, irrespective of whether they are free-living worms or whether they are causing any pathology in humans too, or to animals, or any worm is called helminths. Now, the helminths in the world are divided into two categories based on their cross-sectional appearance. Some are roundworms, some are flatworms. So the flatworms are called platyhelminthes, whereas the roundworms are called nemathelminthes. Now the nemathelminthes you have already learned as nematoda. Now we are today going to learn the platyhelminthes. Now platyhelminthes includes two classes called Cestoda and Trematoda. So what we are going to learn today are the cystode infections. So those are flatworms, which are in lay term are called tapeworm infections. So let's see the classification. Now Platyhelminthes, the flatworms, are in the phylum Platyhelminthes. Now that phylum includes, as you can see here, you can see the Platyhelminthes phylum, and it includes the Cestoda class. Now there are other classes, but today, since we are going to learn about Cestoda, we are going to discuss about the classification relevant to Cestoda. The Cestoda has the order Cyclopaladium. Now this Cyclopaladium has most of the cestode parasites of man and domestic animals, but there is another order called Pseudophilidium, which also has some other cestode parasites of man and domestic animals. That also includes certain species which we will learn later. Now if you take the Cyclopaladium, it has certain families and genuses which are important, which again I am going to skip and come directly to the species here. Now the species that I have listed here, which I have given in these blue boxes, are the ones which are prevalent in Sri Lanka. Now all these species cause human infections, but out of that this H. nana, Hymenelepis nana, is not prevalent in Sri Lanka. So rest of the ones are prevalent in Sri Lanka. In addition, I told you there is another order called Pseudophilidia. Now the Pseudophilidium's order has two other species called Spirometra species and Diphyllobothrium latum, which is the fish tapeworm. Those two species also cause human infections, and out of the two, only the Spirometra species is prevalent in Sri Lanka, and luckily the Diphyllobothrium latum is not prevalent in Sri Lanka, so we are not going to learn that today. So this slide shows you the medically important cestodes found in Sri Lanka. Okay, if I read the names out: Taenia solium, Taenia saginata, Echinococcus granulosus, Hymenelepis diminuta, Diphylitium caninum, Bertiella studii, Taenia teeniformis, and Spirometra species. Those are the cestode infections which are prevalent in Sri Lanka. Now I have also given you the common names used and the stages in man and the animal reservoir, which you can go through later on. And anyway, we are going to learn each of these infections, some in brief, some in detail, according to the level of importance. Okay. So moving on, these cestodes are not found in Sri Lanka, but they are medically important cestode infections. That means they cause infections in humans. Now let's see the general morphology of cestodes. Now I at the beginning told you that they are flatworms. These are long worms mostly. Of course there are short worms, there are only few proglottids as well. But generally they are flat, long, ribbon-like worms. And they have segmented bodies. That means their body is divided into small pieces. Now if you look at the entire worm here, now this is you can see in the picture the entire worm, and if we concentrate on each part of the worm, now they have the head part. The head is called scolex in cestodes, and this scolex is generally microscopic. It's quite small. I mean, the rest of the body of the worm may not be seen to the naked eye, but the head part is quite small. And this head possesses the organs of attachment. Like now, they're generally in the small intestine. They need to attach to the human body in some way. For that, this scolex helps them to get adhere to the surface. They possess various organs of attachment, like the suckers or hooks or grooves. With the help of those organs, they attach to the human body. And then they have the neck region. The neck region is here, which is the region of growth. Now the neck region, of course, this part it's a region of growth in the sense from the back of the neck, a new proglottid is formed and it is being pushed backwards. So likewise, during the lifetime, they produce enormous number of proglottids. So they are being pushed behind by the neck while they are being mature. So closer to the neck, they have the immature proglottids, and in the middle part of the body they have the mature proglottids. With time the immature ones get matured and they are being pushed back by the newly formed one, so the mature ones come to the middle of the chain. And when later on the mature ones, when they get gravid, that means when they get pregnant or when they reproduce and get the progeny going, the latter part of the chain will have those gravid proglottids. Okay, so each of these areas we will discuss. Now this is the scolex. This is how a scolex looks like. But of course, not every scolex will have all these structures. Certain species don't have rostrum or hooks. They will only have suckers. Some of them will have grooves called bothria. For example, the fish tapeworm. They don't have suckers, but they have bothria. So different species have different appearances. But basically, a scolex will have these organs of attachment to make it easy for the cestode to hang on to the human body. Moving on to the general biology, now the scolex has no mouth. Even though it is the head, there is no mouth. So the nutrients has to be absorbed from the host directly. Okay, and the neck, of course, I told you that's the region of growth. So it continuously produce proglottids. And if we move on in the morphology further down to the strobila is the entire chain of segments, entire chain of proglottids are called strobila. Now these proglottids may vary in number based on the species. Certain species like Echinopus granulosus, the dog tapeworm, has only about 3 proglottids, whereas teanid species have thousands to thousands, likewise large number of proglottids in the strobila. And then the shape may vary. Some have rectangular ones, some have square ones, some have cucumber seed shaped ones. So the shape may vary based on the species. And generally the proglottids, mainly we can categorize them into 3 types. I told you before, as immature, mature and gravid. So the immature ones, as I said earlier in the slide, those are the ones which are closer to the neck, newly formed ones. The mature ones are the ones which have developed into the mature stage, that is they have male and female reproductive organs separately in the mature segments. And in the gravid ones, you can see the fertilized eggs. Of course, in the gravid proglottids, they can disintegrate from the main chain and get into the feces or to the lumen, or sometimes even these proglottids can disintegrate and release eggs directly to the feces. So basically, the gravid proglottids have fertilized eggs. These eggs could be released in 2 ways. One thing is gravid proglottids can get break off from the main strobila main chain and come out in the feces along with the proglottid containing eggs, or the gravid proglottid can disintegrate and inside the body it can get dissolved and release the eggs directly into the feces. Okay, so I told you the entire chain is called strobila. So now this is the appearance of a mature proglottid, which contain at least one set of male and female genital reproductive organs. Now, since they have both male and female reproductive organs, they are hermaphroditic. That means they can do self-fertilization. They don't need another cestode worm to do the fertilization. They can do self-fertilization, and sometimes cross-fertilization can happen. It could be between 2 cestodes, or it could be mostly within the same cestode where the chain falls onto each other, like as you fold the clothes, like when they fall into each other, the chain, there can be cross-fertilization happen. And now, even though there are male reproductive organs and female reproductive organs, they share a common genital pore. They don't have different separate genital pore opening, so they share a common genital pore. Now this common genital pore could open to different sides of the proglottis. Based on this side or based on this position, actually it helps to identify the species in certain instances. For example, in species like Hymenolipis diminuta, it opens, the genital pore opens onto the same side of each proglottis, so that when you examine the strobila, the proglottis, you can see that the same side of the proglottis has the genital pore. Whereas in tinea, it alternates, like it could be on the left side in one proglottis, on the other side on the next one. So it alternates, and it's irregularly alternates. There is no pattern. And the fish tapeworm, on the other hand, not the fish tapeworm, the dog tapeworm, Diphylidium caninum, it opens onto both. So this of course shows some of those structures, some of the appearances, how it looks like. You can see the genital pore here. It has opened to one side. It's showing. Now these cestodes don't have a uterine pore. Now uterus in certain other parasites may have uterine pore separately, but these cestodes don't have a separate uterine pore. They only have the genital pore. And the shape of the uterus may differ from one species to another. Now this picture shows different types of uterine appearances, morphologies seen in different species. There may be sac-like ones, there can be branches, there can be egg capsules. So species have different uterine appearances. So with that also we can diagnose or detect which species has caused the human infection. Now this slide shows you different types of eggs that are present in different species of cestodes. Here you can see first the tinea ones, how they look like, and then these are the cestode called Hymenolipis nana, and this is the Hymenolipis diminuta egg, and this is in relation to the size also. You can see the dimension and how it changes. This is Diphyllobothrium latum, and this is Diphylidium caninum, and the Diphylidium caninum generally comes out as egg packets. So these are different aspects, different shapes, and different morphological appearances of different species. But when we learn about each of these, we will go into more further detail. Moving on to the habitat. Now, there are two main life cycle stages for adults and larvae. Out of these stages, the adult usually lives in the small intestine of vertebrates. Okay, the larvae, on the other hand, could occupy tissues of vertebrae and invertebrates as well. So that's all about the general introduction and general morphology of cestodes. In the next series of lectures, we will learn each of these species causing infections in Sri Lanka. And if you have any questions, of course, I know you have no way of asking these questions, but at the tutorial classes you can ask me questions, or when we come back to work, of course you can meet me and ask questions if you have any. Okay, then thank you.